Hello guys, in this video we are going to see about Golgi bodies. Now let's see what is Golgi bodies. It is a membrane bound organelles. It looks like a flattened sac like structures. Its size varies in different species. It is also called as Golgi complex, Golgi apparatus, Golgisomes, dictyosomes, Dalton complex, lipochondria, Golgi material, and internal reticular apparatus. Now, let's see the discovery of the Golgi complex. Golgi complex was first observed by George. Then the morphological structure of Golgi complex was given by Camillo Golgi. So, it is called as Golgi bodies. Then the ultrastructure of the Golgi bodies was explained by Dalton. So, it is also called as Dalton complex. Now, let's see the location of the Golgi complex. It is located near the nucleus and endoplasmic reticulum. Golgi apparatus has two phases. They are cis phase and trans phase. Cis phase is positioned close to the endoplasmic reticulum and trans phase is positioned close to the cell membrane. There is a clear area around the Golgi bodies which is called as zone of exclusion. Golgi bodies occurs in all eukaryotic cells except in mature sperms and red blood cells of animals and in certain fungi, and they are also not present in sperm cells of bryophytes and pteridophytes. This Golgi complex is not found in all prokaryotic cells. Example, bacteria and blue-green algae. Next, structure. We can see the Golgi complex structure in the picture, which shows a stalk of flattened sacs. A complex network of tubules and vesicles is located at the edges of the cisternae. Sort and package these molecules into vesicles for transport to other parts of the cell. Golgi complex is made up of following parts. The first part is cisternae, which is slightly curved and sacs-like structure. They are arranged parallel to each other. It has two phases. They are cis and trans phase. In cis phase, the convex surface faces towards nucleus. In trans phase, the concave surface faces towards the cell membrane. Cis phase is also called as forming phase. The trans phase is also called as maturing phase. The fluid present in the cisternae is called as matrix. The margins of each cisternae is curved. It has three compartments. They are cis, medial and trans. Cis and trans phase are interconnected. Cis phase is present near to the endoplasmic reticulum. Trans phase is present far from the endoplasmic reticulum. Medial is the center layer of the cisternae. This image shows the cis, medial and trans region of the Golgi complex. The next part is tubules, which supports the whole Golgi complex. These are continuous with peripheral edges of the stalked cisternae, but their way of interconnection differs across the stalk. It is a small round tubules formed from the peripheral edges of the cisternae. Next is vesicles, which are small sacs of 20 to 80 nanometer in diameter that develops from tubules. They are found attached to the tips of the tubules at various levels within the networks. Vesicles is of two types. They are coated and smooth vesicles. First, coated vesicles. They have a rough surface and carry the materials from the vacuoles. Second, smooth vesicles. They have a smooth surface and it contains secretory substance. So it is called as secretory vesicles. Next part is vacuoles, which are formed in the concave surface of the Golgi apparatus. These vacuoles are clear and commonly seen at the edges of the complex. It contains a amorphous or granular substance. It also functions as a lysosome. Next, functions of Golgi complex. Its function as a factory in which proteins received from the endoplasmic reticulum are further processed and sorted to transport to their eventual destinations. Secreted biochemicals are later pinches off and passed out through exocytosis. The Golgi complex also helps in transporting the lipids. It is the site for the synthesis of various glycoproteins. In the plant cells, the Golgi apparatus synthesizes complex polysaccharides. Thank you.